Good evening, ladies and gentlemen. My name is Louise Hager, and as Chairman of High Cancer Care, it is my great pleasure to welcome you all to the Natalie Shipman Memorial Lecture 2023. And I'm also delighted to extend a very warm welcome to Professor Ruth Plummer, Professor of Experimental Cancer Medicine, Newcastle University, and to Dr. Adrian Tuchman, Chairman of HI's Medical Advisory Panel, who will also chair the medical part of this evening. Being online has many advantages, much as we'd like to see everybody in person, but being online enables us to reach so many more people from across the UK, Europe, USA, Canada, and of course, Israel, where all our thoughts and prayers are with everybody there. Gosh, I didn't think I'd get so emotional so early. And we pray for peace and that we should all be able to live in peace and harmony. And of course, Susan, Natalie's precious mother, who's joining us from Israel, from Jerusalem. Welcome, a special welcome to you, dear Susan. I just want to bring you up to date. Many of you may not know what's been happening at Chai over the last year. We are currently supporting just over 4,000 cancer patients and their loved ones from age 3 to 98. And this year exactly 1,035 new clients have needed to turn to Chai, which is the largest number ever. And as those of you who heard me speak in the past will know that whenever we quote these figures, it's always with mixed emotions because we just wish there wouldn't be the need. But while there is, Chai is here. There's also been a marked change in the demographic of our clients, with a third of the 4,000 people we are currently supporting now age 50 and under. And so we've extended much more for our younger clients, especially with Chai in Schools program, which is currently providing expert support and advice to pupils, to teachers, to the whole teaching faculty in 29 primary and secondary schools across the UK. And I must make mention of the NHS Jewish Communities BRCA screening program. This is an incredible, incredible program. The NHS put all the resources behind it to ensure that we can reach as many people who may be carrying a mutated BRCA gene. And Chai, together with Genetics, have been awarded the project to inform the community. We had a soft launch, which started in the spring, and to date, over 4,300 people have actually expressed an interest through jewishbrca.org in being tested for the BRCA gene. There's a lot of information on both the Chai website, on jewishbrca.org, and Genetics. So please do look into that. And the hard launch will start in January. So there will be a lot of publicity around that time. So the need for Chai's expertise to support people who maybe have the BRCA gene, test positive for the mutated BRCA gene, will grow even more. And in order to meet our budget, I have to put this in, dear friends, of this year, it's going to be over £3,700,000 to run Chai, to make sure that we can be there to everybody who needs us. We are going to be having a much-standing campaign on the 3rd and 4th of December. Starting with Mad Hair Day in many schools across the UK on the Friday, a Chai Shabbat across the whole spectrum of the community and across the whole of the UK, and the match funding campaign on Sunday the 3rd and Monday the 4th of December. And anybody who's interested and wants to help us reach our target and our goal to make sure that we can carry on being here, please be in touch with Jamie. Any housekeeping notes? The only one I'm going to say is that any feedback, we'd love to hear from you so we can plan future events and lectures that you want to hear. And if anybody would like a CPD certificate, please do email Jamie. Of course, this being a lecture, a memorial lecture to dear Natalie, I'm going to take a few moments to talk about her. And it's almost impossible to believe that it's 33 years since Natalie passed away. Natalie was diagnosed with a brain tumor just before she was 3 and passed away just before she was 8. And even though her life on this earth was very short, her mother, Susan, was absolutely passionate that her short life must be marked in some way. And it certainly has. Susan, together with my dear mother, Frances Weingarten, of blessed memory, were a force to be reckoned with. They created a cancer support organization at a time when nobody talked about cancer in 1990. but they believed cancer had come into their life for a reason and something must come from it. And here you heard just now, 4,000 people are currently receiving support. Natalie isn't physically here with us, but I can say, hand on heart, that her influence wasn't just in those few days, or few years. And in fact, just before she passed away, she knew what her mother was doing. And she said, I want to help. Not only did she help in the formation of the organization, she helps literally every day, because our task and our challenge as we grow and as more people need us is not to forget how we started and how important it is to have the personal touch. And I think we are doing it, because recently one of our clients said to one of our client services managers, I know you support so many people, but every time I speak to you, I feel as if I'm your only client. And that for us is enormous. And how do we do that, dear friends? Over the last year, we provided over 51,000 hours of specialized support. And how do we do that? Through our dedicated team of counselors, therapists, client services managers, led by the most fabulous CEO, Lisa Steele. And so it gives me great pleasure to hand over now to Dr. Adrian Tuchman, CHAI's chairman of CHAI's advisory panel and one of our trustees. Over to you, Adrian.
Thank you, Louise. Many, many thanks. And good evening to everyone, and thank you for joining us. Adrian Tuchman, I'm a retired palliative care physician, and I've worked with CHAI for many years, almost since its inception. And as Louise said, I'm a trustee and I chair their medical advisory panel, who are a team of experts who we're very lucky and grateful to have advising CHAI. And they suggested Professor Ruth Plummer as a speaker, and we're very honoured that she can join us online this evening. It's a very interesting topic that she'll be talking about. Let me just briefly tell you about Ruth. Ruth Plummer, Professor Ruth Plummer, MBE, is a professor of experimental cancer medicine at Newcastle University. She's an honorary consultant medical oncologist in Newcastle Hospitals, at the Newcastle Hospitals NHS Foundation Trust. She directs the Bobby Robson Cancer Trials Research Centre, and I think she'll tell you a little bit more about that when she talks. She leads the Newcastle Experimental Cancer Medicine Centre and CRUK Newcastle Cancer Centre. Her NHS practice is in skin cancer, treating all types of melanoma with systemic treatments, which she'll tell you a little bit more about. And she was awarded an MBE, which shows you how prestigious she is and how lucky we are to have her at the Queen's Jubilee Birthday Honours for Services to Medicine. So, Ruth, thank you so much for joining us, and thank you for talking about a topic that I'm sure is going to generate a lot of interest, taking new cancer drugs to the patient. And if I can say to the people who are on the call, if you have any comments and any questions, we'll do our best to answer them, hopefully all of them, but maybe some of them at the end of the meeting if we have time. Please put them in the comment box. Thank you very much. Ruth, over to you.
Thank you, Adrian. And it is a huge pleasure to give this talk. And I mean, I've read about the incredible work that CHAI does, and I was hoping to try and explain to you what I've been trying to do over the 20 years or so that I've been a consultant in Newcastle. And I was going to talk about taking new cancer drugs to the patient, because that is what I do. Interesting. Oh, there we are. I briefly lost my little pointer, but I found it again. So I was going to talk to you just to quickly summarize where we find new drugs, and then talk about early phase trials, which is what I do, because cancer trials are different in this space. And then a little bit about academic drug discovery in Newcastle. And this is a really easy group to talk to this, because the first drug that Newcastle developed, cancer drug, the first Geordie cancer drug, was actually the first PARP inhibitor. So this speaks very much to what was being said earlier about the BRCA story. But then also talk about the importance of working in partnerships to do this. So where do we find new cancer drugs? And I hate the concept of the war on cancer when people talk about having a brave battle, because of the you know, the concept of losers and the fact that people are, you know, are being put in the position of feeling as a patient they have to win the fight. But I do use it as an analogy because there are, in effect, the three forces when we're thinking about cancer treatments. And the surgeons, my surgical friends and colleagues, know they are the senior service because they've been trying to treat tumours with surgery for many years. And that's an amputation kit from the 1800s. And doctors have tried to cut tumours out. Radiotherapy is really interesting. About half of cancer patients get radiotherapy at some point. Radiotherapy now does do trials, but didn't used to. So Röntgen discovered X-rays in 1897, and within about 6 months, X-rays had been used to try and treat tumours. Röntgen, when he discovered these weird things he called X-rays, the first X-ray he ever took was the door, and the second one is that picture there, which is his wife's hand, and it's his wife's left hand because you can see her wedding ring on her finger. So, but they're the cavalry. And the reason the analogy is useful is that medical oncology, there aren't very many of us medical oncologists, we only do cancer drugs. But cancer drugs have only been around since the 1940s, 1950s. And the first cancer drugs were actually developed out of the First World War, and sometimes out of terrible things, good will emerge. And it was the nitrogen mustards, the mustard gases. And it was noticed that people who survived being gassed dropped their blood counts. And in Knight's move thinking, doctors in the States thought you might be able to use that to treat leukemia. So we very much, medical oncologies are the junior service, and there have already been cancer drugs since about the 1950s. Where do we find them? Well, there were the nitrogen mustards, and actually we've done an awful lot of finding natural products. On here are the periwinkles. Periwinkles, there are natural poisons. If poisons damage human cells, that's what cancer doctors who are using drugs want to do, damage the cells that are wrong and out of control. So there's an American oak apple there for etoposide. There's the periwinkle. There's a picture of flame in the forest on the yew tree in my garden, because yew trees, two very powerful cancer drugs have come from the yew trees. But also nowadays, we do what's called rational drug design, where we look at the proteins, the things that have gone wrong in the cancer cell, and then try and alter them by making molecules, sometimes in silico, as in on the computer, and then doing the wet chemistry, as it's called, to make a drug. So that's where we find new drugs, and there's lots of them. It's really challenging, and one of the good things I've seen in 20 years is this timeline coming down. This is a slide from the Association of British Pharmaceutical Industries, ABPI, talking about the challenging timelines of drugs. Not just cost, but the number of molecules to get one good drug out the end, but also the time. And I do clinical development of drugs, and that has traditionally taken up to 6 to 8 years. And then there's the regulatory review. So you hear about a breakthrough, but then everything seems to disappear as we very carefully do the safety studies. And that's what we do to take drugs into the clinic. And I said early phase cancer trials are different. And since the pandemic, it's been easy, because tomorrow morning I'm doing my phase 1 clinic again. It's been much easier to explain to people, because everybody heard when we were waiting for the vaccines to come through that you had the safety study, you then had the phase 2, and we needed to wait for the pre-registration phase 3 randomized trial. In non-cancer medicine, studies that work out dosing schedule, try and work out the side effects, explore the toxicity, because you won't understand everything at that point, are done in healthy volunteers. But you can't do that in cancer patients. And that's because, as oncologists, what we want to do, as I alluded to before, we've got cells, we're trying to treat within a patient, that patient's cells that have gone catastrophically wrong. And to treat them, you actually want to kill them. You want them to go away. So we have never done healthy volunteer studies, and the patients I see on a Thursday morning know they've got cancer, know they've run out of standard treatment options and will die of their cancer, and are prepared to take something experimental. And I very much regard them almost not as the team because they're patients and we're looking after them, but they are really contributing to research. And we used to, because I grew up with just chemotherapy, we had a very straightforward mathematical design. We wanted, as you know, those of you who've looked after loved ones or experienced chemotherapy yourself can guess what we did. We thought more is better. So we would start with what we thought was a safe but not too low a dose, because we're treating patients, and treat 3 people, and no bad side effects happen. We used to actually double the dose and then treat another 3. And if one patient got bad side effects, you would add some more to the cohort, traditionally another 3 more. And if it was only 1 out of 6 people had bad side effects, you'd go up in smaller jumps, but you'd keep going up. And as I say, people for chemotherapy, and I am sorry, because I think my slides, since I last looked at them, have got slightly scrambled. So I'm sorry about the text, or the text I can see certainly doesn't look like it was. But anyway, we used to go up to what we called the maximum tolerated dose, which was the biggest dose we could get away with in most patients. And that's how we set the doses of cancer drugs. Things have changed. In the early 2000s, there was a really seminal paper by Hanahan and Weinberg saying, what are the hallmarks of cancer? What goes wrong in those cells? What I think, and what I'm showing on this slide, is they updated it 10 years later. They added a couple more hallmarks. But what was really interesting is they also pointed out that over that period, we'd ended up with drugs to target most of the hallmarks of cancer. Very different mechanism of actions from just poisoning cells with chemotherapy, which does mean we've got different toxicities and different ways of having to think about and do our trials. But it has meant that we've got far more drugs coming through. And I'm going to talk about PARP inhibitors later. And you can see that here, they are one of the things that will target the hallmark of cancers. It's also meant the trial design, for me, has got more interesting rather than the mathematical 3+3, because you can answer now with the regulators lots more questions. So this is an example of an ATR inhibitor, which is another DNA repair inhibitor, like a PARP inhibitor, where you're trying to control what's gone wrong in the cancer cells. But it was done in combination with chemotherapy, with a PARP inhibitor, and with immunotherapy, all within one trial, lots of parallel cohorts. It's much more interesting for me when it's a wee bit more complicated, the trial, and you've got other things to think about. So we have evolved our trial design over the years. And the other thing that is changing, and is absolutely changing it again, is precision medicine. And it was talked to you earlier about, looking for BRCA, but we are now able to look for, in our patients, for the things that have gone wrong in the tumors and target those with some of the newer medicines. And the poster child for this is larotrectinib, which targets a rare mutation. And the developers of this over in the States did a study where they recruited from children right up to 90-year-olds, so over the range that you talked about, the higher supporting, if they had the mutation. And this is a waterfall plot, much loved of cancer doctors, where when you put a patient on a trial, you measure everything on the scan and make that 0.0. So if the tumor grows, it would go up. And if it shrinks, which is what we all want to see as oncologists, you see this waterfall. These are individual patients with the tumor shrinking. Some of them, it's shrinking down to 100%, a complete response. And a swimmer plot shows these, again, are individual patients with months along the bottom, how long people would stay on the drug. This was a basket study. So it didn't matter where your tumor came from. If you had the mutation, that was in the basket and you got the drug. We also in the UK ran from Birmingham a really important study called the National Lung Matrix, which was an umbrella study because it was all lung cancer. But the lung cancer was profiled with initially a few genes, but it ended up with 40 different genes. And then if you had a particular gene, you went into a different spoke of the umbrella. So it was all lung cancer under the umbrella, but the different spokes were individual groups that were being tested depending on their tumor. And this is changing the timelines. This is what's been one of the things that's been great to see, in that there was a drug called crizotinib developed in 2005. 2 years later, the fusion that it can target was found in lung cancer. 2 years after that, patients had been treated, and 2 years after that, it was licensed. So if you know where you're going, maybe it's obvious, you can treat a tumor and you can get a drug to license and marketing and therefore worldwide availability much more quickly. And what we're doing in the UK at the moment, and the Target National Study is jointly funded by the Christie Hospital Charities and the Sir Bobby Robson Foundation in Newcastle, but it's running across all 17 experimental cancer medicine centres, where all of the patients referred to see us to talk about trials, we can do on a blood test, we can look at circulating tumour DNA in the blood and look for changes that we can then match to a trial. And I was on a call this afternoon for an hour and a bit with the rest of the country, looking at what we've seen and using everybody's knowledge. Where are the trials? What can we offer and what can we access to patients with mutation? And Cancer Research UK has worked with NICE to set up a study called Determine, where, including for pediatric patients, patients with can have access to a licensed drug in an indication where it's not yet licensed, to try and build the evidence that this drug will be good for a wider group of patients. And so that this is what we're doing for our patients, as well as other studies we've got open. I also said I was going to talk about PARP inhibitors, because Newcastle was the second place in the UK where CRUK, well, it was CRC at the time, Cancer Research Campaign, set up an academic drug discovery unit. Two mentors of mine moved up from the Royal Marsden, the Institute of Cancer Research, to set up this second unit in the north of England. And that's where I've been working. And there's now been 40 years of cancer research in Newcastle, from when CRUK invested through all the way through, with now the Great North Children's Hospital and our new cancer hospital, and amazing facilities we've got to look after patients, but also to offer them trials. We are a CRUK centre, as Adrian said, and an ECMC, where those two big programme grants reckon that Newcastle does is the sort of early phase trials, and also clinical pharmacology and drug discovery. And clinical pharmacology is looking at drug levels to help with safe dosing, and drug discovery is drug discovery. I also, this is my catchment area for recruiting. There's the Scottish border, there's the North Lakes, Manchester down here, Glasgow up there. So Newcastle, Manchester and Glasgow are, in the top 5 most active early phase trial sites. So it's a big, big triangle in the north where we share a lot of data and a lot of patients. I always say, when I'm talking in London, I point out that it's the nice bits of England as well. I'm not allowed to say the nice bits of the UK because my Scottish friends get cross with me. But I was saying earlier to the team when we're having a rehearsal, I've been unable to change my slide of the tree in Sycamore Gap. I guess I'm going to have to change my picture of beautiful Northumberland, but not yet. We have an amazing setup in Newcastle. We are at the Northern Centre for Cancer Care. We actually now treat North Cumbria as well as Newcastle patients. We have an outreach centre over there, and we've got the Great North Children's Hospital, but amazing research laboratories as well to support what we do. And this was a picture of, and it's certainly lost the background, there was a picture of Newcastle, and I could have shown you where the football stadium was, but actually it's all within I shall skip over that. I don't know what's happened to my slides, but it was to show you that it's all within a mile or so. So this is very easy for us to be joined up and to work together. And my colleague who does this with me now, Steve Wedge, is a cancer drug discoverer. And then I'm at the other end of this when we take the drugs into the clinic. But we need scientists who work out what's gone wrong with those cancer cells to find the target. Newcastle doesn't have compound collections. We ask other people to screen for that, but we do have structural biologists who design drugs. We then have medicinal chemists who also design drugs. And then you need to optimize your drug and to check it works. And all drugs, in terms of their toxicity, are tested in animals before they are given to a human. And we do the clinical evaluation on the Bobby Robson unit. And Newcastle has 2 licensed drugs now that have gone into the clinic, erdafitinib and rucaparib. It's a bit of a long and winding road. This is the Lake District. I was not supervising the small child off the path at this point, and my twins are here, and I was definitely not the parent in charge when that photograph was taken. But I was saying, this is a tortuous road to do cancer drug development, but it's better working in partnership. As I alluded to, we had 2 drugs come through for research in patients in Newcastle. Rucaparib was the first PARP inhibitor into the clinic before olaparib, a couple of years before olaparib, but is a Cancer Research UK drug and an academic drug development. And also there's a drug now licensed in bladder cancer, erdafitinib, developed by the Newcastle scientists and chemists. But I was going to talk about rucaparib because in 2003, I wrote the first prescription for the first cancer patient ever to be given a PARP inhibitor. And that was one of the sort of seminal moments in a career I've had that's been a huge privilege. And PARP is Adrian said I wasn't allowed to do too much science, but it is a dead cool enzyme. We have PARP in virtually all of our cells. And there's about 1 PARP molecule in the nucleus, PARP1, for each kilobase of DNA, and it's there to look after our genome. It's really important when our cells divide that they copy our genes accurately. But during the average day, because of the cells doing their metabolism, you get things called reactive oxygen species, which can damage or make little nicks in the DNA. And when PARP sees a nick, it binds to it, and it activates it, and it makes a polymer of ADP ribose, hence its name, PARP. And it puts the polymer on itself. And because it's bound either side of a little nick in DNA, you've got 2 negative charges because the polymer's negatively charged, and it just flexes it apart and allows the DNA repair mechanisms to pop in and mine to repair this. Now, where PARP inhibitors started in the clinic, it wasn't the BRCA story. And I was going to start with a story on this because for me, the clinical story, as ever, starts with a patient. And the second person ever to get a PARP inhibitor was a painter and decorator called Peter. When I met him in 2003, his children were teenagers. He had an inoperable tumor, and here's a CT scan with his tumor marked on it. And he'd run out of options. His only other option, he'd been told, was symptom control. So he was prepared to take a drug where we said we weren't sure what dose, we didn't know whether it would work, and we didn't know what the side effects were. And those are the people like Peter I'm going to be seeing in clinic tomorrow morning. But the story of PARP inhibitors in cancer medicine actually started in Brighton. So I'm not going to be too north of England focused, but with a lady called Barbara Durkatz working with Sidney Schall, who were there. And Barbara, this was before the enzyme was found, the protein that we were able to model and drug. But she noticed there was this funny reaction of making these polymers of ADP ribose happened when there was DNA damage. And she had a weak PARP inhibitor and discovered that if you block that, you got more bang for your buck. And this is from Barbara's Nature paper in 1980, where this is her dose of chemotherapy drug along the bottom, killing the cancer cells in a dish. And she could kill more cells with the same dose of chemotherapy if there was a PARP inhibitor. And that's actually where the PARP inhibitors started. And rucaparib was the first Newcastle anti-cancer drug. I have shown a structure here, partly as a homage to one of my friends and mentors in this, the chemist who made rucaparib, Roger Griffin, who sadly died of pancreatic cancer before his drug finally made it properly through the clinic. But Roger said to me, if I ever did a talk without a structure in it, it was boring and all the chemists would leave the room. So I always have a structure in my talk. But the reason for putting it here is I said this was a dead cool little enzyme. And what PARP does is it takes nicotinamide, which is a compound we have in our cells, breaks the nicotinamide bit off, and this is the ADP ribose, the negatively charged bit. But the reason for showing you is this is the bit that binds on the enzyme to block it. That needs to go in there. And all of the drugs have got that initial shape, and then the chemists made them more complicated so they bind and stick and actually stop the enzyme working. And this was Barbara's compound. In 1990, CRUK invested in drug discovery in Newcastle, and this was the first project. And 2003 was when we treated Peter on the trial. And we thought that we were going to make chemotherapy better. And these are our slides, and I'm not going to go through them in detail, showing that we could poison, like Barbara did, cancer cells better with our PARP inhibitor. We could actually make radiotherapy work better. So this is what we thought we would do. And we did a trial, first in human, first in class, with a PARP inhibitor. And I went to the ethics committee and said, these drugs, there's no science that this drug would work on its own. So we need to do a combination study with chemotherapy. We'll give one dose on its own to get drug levels, check it doesn't do anything bad, but then we're going to give it with chemotherapy. So this was the design of our trial. And our patients very generously, some of them allowed us to take biopsies of their tumor as well as blood tests. And we knew straight away the drug was doing what it said on the tin and blocking the PARP enzyme in the blood. But also, this is in the tumor. We had a baseline biopsy, and this patient, there's only one patient here at one of the low doses, but in his tumor, the drug was getting into the tumor and it was 80% inhibited. This is Peter's tumor after 12 months of treatment. After 20 cycles, 20 months of treatment, I suggested to him that we looked at surgery for him because I was worried about the amount of chemotherapy I was starting to give him while his tumor responded. But he had a dramatic response to treatment. But at the same time, and this is where my slide builds, I should have not put builds in my slides because they're not working tonight. The science moves on, and it showed how wrong we were in that the story came out both from Newcastle and Sheffield scientists and the ICR that PARP inhibitors would work on their own in patients where the tumor had the BRCA defect. The paper came out in Nature, paired papers in Nature from Newcastle and the ICR in May 25. And 6 weeks later, I'd gone back to CRUK and said, not quite, we were wrong, but we've had a better idea about how we take this drug forward. And can we do a phase 2 study, because we knew a dose, in BRCA deficient breast and ovarian cancer? And again, the second patient we treated on that responded. I don't need to tell this audience about the importance of BRCA, but when I'm talking to school children and the medical students, I highlight the fact that there's some very high profile people have highlighted the importance of knowing and understanding about having a BRCA defect because of the high risk of lifetime risk of cancer. So we'd said they wouldn't work as single agent. We were completely wrong. And it was Thomas Halliday at Sheffield at the time, now back at the Karolinska, who said this, because I told you about all those little single strand breaks that happen, those little nicks. Normally PARP just repairs them. But if you block PARP, they don't get repaired, so they're sitting there. If the cell then needs to divide and copy its DNA to make 2 cells, which is what tumor cells are doing out of control, it's like pulling 2-ply wool apart. It'll stay together while it's twisted, but if you pull the strands apart, you'll get a break. And cells really don't like double strand breaks right across their DNA. For most of us, that's not a problem because we've got double strand break repair, one of the most powerful DNA repair pathways to protect our genome, and it gets repaired. But if you've lost double strand break repair, then the cell is stuck because it's got these single nicks that are just happening and they can't be repaired, and then the double strand break comes as it peels off. BRCA1 and BRCA2, those 2 proteins that are mutated in the inherited breast and ovarian cancer tendency, sit in the double strand break pathway. So Thomas said, if you think of patients with a germline BRCA defect, they've got one good copy in most of their cells, but the tumor cell has lost the good copy. That's what goes wrong. So it can't repair. If you block give a PARP inhibitor, it's what's called synthetically lethal because you've got 2 genetic defects that cannot work together. Now, when you've got a good idea, lots of other people join. So this was the drug that we started with, AG 014699. I shall never forget the number, that went initially intravenously and then orally into cancer. But AstraZeneca started 2 years later. Well, kudos, the small company that AstraZeneca bought out and developed Laprib. And there are the other PARP inhibitors, and I now can't fit how many there are onto my slide. We, when developing rucaparib, showed, like Olaparib, that startling response rate. This was the Ariel 2 study. It's the waterfall plot presented by a good friend of ours, Professor Ian McNeish. We were at medical school together, and Ian works at Imperial now and treats ovarian cancer, and he led this study. But look at this in patients with a germline or somatic, which means just lost both BRCA in their tumor by chance, it's an over 80% response rate to rucaparib, and similar to the other PARP inhibitors. So these are really powerful drugs if you've got that genetic defect. That Ariel 2 data meant that the FDA gave rucaparib was the first PARP inhibitor that was given breakthrough designation. Clovis Oncology, who were developing it by this point, because academics can't afford to do the expense of all drug development, so you always end up with an out-license or an industry partner. But they sent the trials unit where we treated the first 3 patients, whoever got them. When they got the breakthrough designation, they sent us a thing, and I was really pleased because they etched the structure on, so it was there for Roger. But also Cancer Research UK gave the Newcastle team their first ever translational science team prize. This wasn't a prize for a person. This was a prize to show that this is a team. You can only do this with a team. It was a bit of this is our development story from 2003 through to the licensing in 2016-17. It was a little bit of a story as well. So we were ready to go into the clinic. I said it was a long and winding road in 2001, but we'd partnered with a small company called Agaron to help make the drug. But they were acquired by Warner Lambert, and Pfizer acquired Warner Lambert, and all of that due diligence, as it's called, meant it was 2 years. We would have been in the clinic in 2001. In 2008, AstraZeneca and Pfizer deprioritized their PARP inhibitors and said, actually, these drugs don't work. They're not good enough. We kept pressurizing Pfizer that this was potentially a good drug. So one of their senior medics said to me, write the killer trial. And that was the trial that Ian McNeish helped us to write that became the Ariel 2 study. And a year later, Pfizer said, well, we've given up on this drug, but Clovis are prepared to take it on. And Clovis were really good as a company, because they came to Ian and I said, we like that trial design and we'll let you do it. And that's how we managed to get rucaparib over the line. What was lovely for me, in 2018, the university and CRUK decided to have a celebration event. One of my learning points in life, in that I had skipped an executive board meeting because they were always boring, and noticed in the minutes that this had been decided and I was organizing it. So I now know not to miss key meetings. But anyway, what's lovely for me is that we held it in the Great North Museum. Everything in Newcastle is Great North, like Great North Rum. But we had a public engagement event in the morning and a scientific symposium in the afternoon is what we organised. And incognito, Peter and his wife came along to hear the end of the story. I said, do you want to hear what happened with the drug that you were prepared to take when we didn't know so much about it? And we ran this just before rucaparib was given the European license, and soon after that it was NICE approved. So I said, and to wrap up, that it is partnerships that drive this. And we do work with industry. We can't afford to do this on our own. And I have a lot of friends and colleagues in industry. And actually at Newcastle, we now partner very closely with a company called Aztecs, which is based near Cambridge. And not only do they invest in staff at Newcastle who do drug discovery with us, but they've actually, one of my friends and colleagues on the phase 1 unit, Christoph, is seconded 2 days a week and works back at Aztecs, training in industry how you take a drug into the clinic so we can learn to do it better. The other major partnership, as is clear, for all cancer treatments, whether it's the support that HI gives or the research thing, is the incredible charitable generosity, particularly in the UK. If you talk to European colleagues, we have, as a nation, a real tradition of charitable giving. It is fairly well known that Sir Bobby Robson had melanoma, so he was my patient because he started his life in Durham in the north of England, and his wife told him it was time, when he was starting to retire, he never really retired, to come back home. So he ended up in the northeast, which is how he was my patient. When we were raising money for the unit, I was asked to ask him if he had any business contacts who we could ask for a donation. And he decided to set up the foundation. And this is a photograph of an event we did together, which he then sent me in the post saying, "Why did you ask?" which was a joke he'd made when he and I were forced to learn charity law as we set all of this up. But so I'm very proud to lead the Sir Bobby Robson Cancer Trials Research Centre. This is Lady Elsie coming to one of our public engagement events. We hold them. This is my lab team teaching people. We held a couple of weeks ago for the students in Newcastle, to the pharmacy students, the nursing students, to show them what clinical research is and get them interested. Since we opened and Sir Bobby opened us, we've had an increasing number of trials and see more than 1,000 patient visits per year through the unit, even though we only do those very early phase, very specialized trials. And we are absolutely planning for the future because we've got to capacity. We're turning down about 1/3 of the trials we're offered because physically can't fit any more desks or any more people or patients in the unit. And what's more important is we have a waiting list of patients who would like a trial and are saying, well, and the patients I see often say to me, well, if it doesn't help me, it might help somebody else. And that is an incredibly generous thing that people do at the end of their journey to help us work out dose and side effects of new cancer drugs. And we have a waiting list of people wanting to do that. So currently having discussions with the Trust University and the foundation, we've got a mock-up of, it's currently being a car park, and the nurses keep an eye on it for me that nobody builds on what we think is our bit of car park, to see if we can get a new and bigger building, which would be the next stage for us for planning for the future. So what I hope I've shown you is that it is complex, it's expensive is the problem to do it safely in the clinic. So we can't do this purely academically. So we do work with industry, and that partnership can be really successful, stimulating and fun. In the last 20 years, we've seen a huge increase in flexibility on how we do trials. And I actually think that the joint training opportunities with the foot in both camps that is now really acceptable as we train is the way we will bring more benefit to patients. And with that, I was going to finish and say thank you very much for listening.
Ruth. If there was an audience, they'd be clapping you. So I'll do the clapping, Ruth. So thank you very, very much indeed. Fascinating and insightful. Excuse me. If anyone would like to ask Ruth some questions, please post them in the comment box. Maybe I'll start a little bit of sort of a chat with you, Ruth, whilst we're waiting for a few comments. And I know what the answer is, but I'd like to hear it from you. Your advice when people are offered to be in a clinical trial, because people get very nervous when they're asked. I know some people say, I want to do it to help others, but some people get very anxious. Your advice is?
Ask the question. So we're a bit obsessed with informed consent and have really tedious, long information sheets, but encourage people, our patients and their families, to take them away and read them, but also to ask their GP, ask their family, and come back with a list of questions. I I actually spend quite a lot of time on Thursday mornings saying to people, with people saying to me, I've got nothing to lose. And I saying, we're going to ask a lot of time. And none of us know how much time we've got, but some people have got a very big warning sign that time is limited. And they have got things to lose because we might give them side effects, no benefit. So we might gain knowledge, but they would give. And so it's not that I discourage people from going on trials. I try to be honest about the choices, but it's informed consent. I think there are the later phase trials where there's more data, more promise, but they don't always work. And everybody, when you have a late phase trial where there's the standard arm, patients always want the experimental arm. But if the experimental arm was always right, we wouldn't need to do the trial. So trials, it isn't, there has to be a question, or lack of equipoise is how you would put it as a doctor, in that you don't know. You're asking a research question and you have to be honest with your patients what you don't know. It's just that with a phase 1 doctor, there's an awful lot we don't know.
Yeah. And if you do know, then you're advised to, I get it. Again, there may not be an answer to this one, but people listening to you are hearing there's some new stuff out there. From apart from the old traditional chemotherapy drug, there's new drugs out there. I want to be in a trial. How do they access trials? Because obviously people are very limited in what they can access, usually according to the treatment center that they're being treated in.
Yes, because not all trials are open everywhere. There is a big push to improve equity of access and for patients to be able to access research, but it might involve travel because not everywhere has the facilities to run all trials. I mean, I'm saying we haven't got the capacity to open them with all the regulatory things we need to do. So it is tricky. But there are now much, much better information sources to look at this. The CRUK website is excellent. To be fair, the clinical nurse specialists often know exactly what's going on in the region. They're fantastic support to patients who are interested. But you only want a trial at the right point, because if there's a good standard of care, you have it first. Nothing's a guarantee, but if you know what the response rate is, that's much better. I can see in the chat that somebody's saying, do you treat people like experiments? Well, I try very hard not to, but I'm very honest that we are trying to work out dose and side effects. And with cancer drugs, the only way to do that is in patients. And so it's not an experiment, because I work with nurses and doctors, and somebody asked me once, what am I proud of? And it isn't having an MBE or having taken a drug into the clinic, etc. It's actually those patient stories. There's a lady who happens to have a BRCA defect who went on to a PARP study and said to me, I'd really like to see the kids through primary school. Her children are going to finish school this year. And that really matters.
Yeah,
that's what the fact that they've been, and she actually had olaparib before they got the formulation right. She took a chance and went on a trial and got lucky.
Yeah,
she didn't have it as standard of care. She got lucky on a trial, but not everybody gets lucky on a trial. And I try really hard that we don't offer false hope.
Yeah, and that term "lucky" is a difficult term to use. So if I can just sort of go through a couple of the questions that have been asked. So Ingrid asks, what about blood cancers like multiple myeloma, where there are no tumours, lumps and bumps? Yep.
Well, the hematological doctors have always had it easier in terms of their research than we do, because there are no lumps and bumps, but the cells are circulating in the blood. So they've actually been able to access sequential biopsies in a way that we can't do with solid tumors, because you can keep doing the blood sampling. And what's transformed for us recently in terms of matching patients to trials and understanding the tumors is circulating free DNA, which is allowing us in lumps and bumps, solid tumor doctors like I am, where, because all of our cells, as they turn over, you get little fragments of DNA in our blood. But if you've got a tumor on board, it's a mix of DNA from the tumor cells your normal cells. And they now can sequence that to such level of detail as what we were discussing this afternoon. They'll say, tumor fraction, 2% of the DNA, and we've seen X, Y, and Z mutation. So we're actually, the hematologists have been able to do this sort of research, and it probably, not only are a lot of the leukemias childhood tumors, and those, because they tend to be one catastrophic thing, it's been chemotherapy, but leukemia, childhood leukemia, has a reasonable cure rate.
Yep.
I mean, it's not good enough, because I don't think anything ever is good enough, but it is better than the solid tumors have ever had, because of the way you can actually understand and access the tumor cells.
Yeah. And I'm going to ask the next Joan's question next. So, have drugs which were in use 10 or 12 years ago gone out of use now, e.g. paclitaxel? Again, it's just a general question, really.
No, they're sometimes bizarrely challenging when they go off patent, because somebody still has to make them. So there've been weird shortages of some of the standard old-fashioned chemo drugs, but loads and loads of patients get paclitaxel. Paclitaxel
right.
You can make now. It was found in the bark of the Pacific yew tree, so it was very bad for the trees. Docetaxel, the one that's used more commonly in breast cancer, is from the leaves of the European yew. So some of the big National Trust places used to be able to harvest and pass on their yew clippings to extract the drug. We all knew yew trees were poisonous. Well, they're really very poisonous.
Very, very. And Lawrence is asking, I've been off chemo for 4 months, had CT scan result tomorrow. Is it normal to wait for so long for a CT scan? It's a general question. Can you take chemo tablets? I'm not quite sure I understand that, but yeah.
So it slightly depends what the aim of the chemo was and what it's for. Yes, we would scan every 3 months generally, and when somebody
Yep.
stopped chemo, I would scan 12 weeks later. So it's roughly right. Yes, in that I guess if you look too often, you won't know what to do as well. And sometimes tumors, when they're responding, can swell a bit. So you need time to check you've got it on the right trajectory. You can take chemo tablets. So there's a drug called capecitabine, which is there are a number that are tablets, slightly trickier, and your bowel doesn't like it very much, because I guess, as the tablet dissolves, you're getting a chemotherapy dose to the lining of the bowel. So they can be quite tricky in terms of GI symptoms and side effects. But you can do it. Yes, you can make things into tablets.
And that's again helpful for Lawrence, I'm sure. And a lot of the talk we get, there's clearly been big changes, and you implied this over the last 10, 15 years. Genomics has made a big difference. The non-chemotherapy cancer treating agents that are getting that are out there. So cancer, this is a question, is it being transformed into a sort of more of a chronic illness for many people? What's your view?
Well, my view is sort of two ways. One is I hate it when I don't like the cure word, because I don't like to use it, because you're making a very big promise and you don't know. And I guess I know from years of experience that this is a beast of a disease that just comes out of the blue back again. But I think it is quite possible now some of the patients I've treated with melanoma with immunotherapy may never need treatment again. I don't know yes. So but what does cure mean? It means that, in effect, you die of something else. You live your normal life expectancy. I think we are starting to do it. It's a bit of a tough chronic disease, but then cancer physicians nobody else goes around saying, when are you going to cure diabetes? When are you going to cure heart disease? But it's
right.
because of what we're treating. So cure is not held over other physicians, but I absolutely can see why that is what you want to get it to go away. It's tricky, but I think with early detection and some of the powerful new immunotherapy, I think there are patients that we will get to a point where they're not needing treatment. And I've got some melanoma patients who've been off treatment 8-10 years now.
Yeah. And that's really, really helpful to hear. And I think it is a good sort of statement to close the meeting that we've had today. So Ruth, I want to thank you, really thank you from here. It was a great talk, very informative. It'll open up people's eyes to the new world of cancer treatments. And we are truly very, very grateful that you could join us this evening. Ruth, thank you very much. If there's anything we could do to support you, please let us know as a charity. And can I also thank the audience who have attended this evening, and thank you for your support. Ruth, thank you.
No, absolute
Good night.
pleasure. Good night.
Good night. Good night, everyone.